Serological methods. Now we will study the virus neutralization test. It is also the similar, just like we have seen the serum neutralization test. It will be, of course, the serum. For serum we are using the serum with the neutralizing viruses, and here we will see the virus neutralization test. We will neutralize the virus in the different ways. Serological method that detects the presence of viral neutralizing antibodies. Of course, we need those antibodies which are going to neutralize that virus. We will use specific dilutions of the serum to neutralize that virus. The antibodies bind to virus particles. So which kind of antibodies? It may be the primary antibodies, it may be the monoclonal antibodies, or maybe the polyclonal antibodies which are going to neutralize that virus. And after the neutralization we can see that what kind of virus is this. It blocks the viral infectivity and no cell infections occurs. It is conducted by mixing dilution of the viruses. So we will see that how the virus neutralization occurs. We will use the different types of the dilution of the serum, and we will use those different dilutions with the different dilutions of the viruses, and we will dilute them, and so that the virus will not cause any infection after using these antibodies. Incubating them for culture into the cell. After we using the dilutions and we will making them together, we will incubate it and culture it into eggs or animals to have the clear cytopathic effects. The virus neutralization test also dependent on the cytopathic effects. It means that we need to use some animals, and then we can use it in the cell lines, and we can see the cytopathic effects. Just like you can see in the diagram, this is the virus, and then this virus will be entered into the different dilutions of the serum. The antibody is the serum is the same thing, antibody molecule against the virus and attachment size. If the virus will be neutralized the antibodies, then it will be cause no infection. Then we can see the cytopathic effects. There are two kinds of the cytopathic effects will be clearly seen in the virus neutralization test. First is the rounding of the cells. The cells will be rounding by different ways and change in the texture. It may be granular or hyaline or the glassy texture. The cells of the structure of the cells can be the glassy, or its texture will be the change, or it may be we can see the formation of the syncytium. It is always there, which is also an important cytopathic effects, and we can see that virus whether the antigen and antibody reaction occurs successfully or not. The procedure for the virus neutralization test, we have some different steps which can be used for the virus neutralization test. The first step is the serum dilution of the serum. Of course, we will use the different dilution of the serum. Maybe it is 10 into raised power 1, 10 into raised power 2, or 3 or 4 different dilution depending on the virus, and then continue towards the maybe the 10-fold dilutions of the serum. And the known virus will be known and incubated for 1 to 2 hours at the 37 degrees centigrade. These are the known of the serum solution, and which will be suspended inside the virus. Second step is cell culture inoculated with the mixture, and it will be incubated at the 37 degrees centigrade, and then we can see it under the microscope whether the plaques formation occur or not. The pros and cons of the virus neutralization test. It can be specific, of course, because we are using the antigen and different dilutions of the serum, so it will be very specific antigen antibody reaction, and it is very sensitive because different dilutions of the serum has been used, and it is used for the vaccine production and immunological status, because we are going to neutralize the virus. So if some specific kind of the antibodies which are going to neutralize that virus, they are very successfully can be amplified for the vaccine production and vaccine preparation. Those antibodies and that antigen can be used as the vaccine. However, it has some of the disadvantages along with the pros, that this is very slow process. Of course, you are using the different dilutions, and then you are using in the viruses. It is intensive, required skilled people, because we are using to deal with the live virus, and also it is dependent on the different kind of cell lines which we are going to use in the experimental purposes in the cell culture.